The U.S. Midwest is an extremely productive agricultural region. During the crop growing season, photosynthetic activity of corn and soybean in this intensively managed critical zone rivals that of the Amazon rainforest. But in the late fall to early spring, this same region is nearly barren of vegetation. In the Sangamon River Basin in central Illinois, we measure this vegetation activity or lack of activity at an eddy covariance flux tower. The carbon dioxide in the atmosphere is taken up by crops over the course of a growing season through the process of photosynthesis, shown here as red arrows. This carbon, called gross primary production, is used by the plant to increase its above and below ground biomass in the form of leaves, stems, seeds, and roots. At the same time as plant stomata are taking up this carbon, they are also releasing water vapor to the atmosphere in a process called transpiration, here shown as blue arrows. The cumulative transpiration over a growing season is illustrated by this cloud of water vapor. Transpiration and evaporation are important parts of the water balance in addition to rainfall and runoff. Carbon uptake and transpiration both vary with growth stage and crop type, and they both depend upon and influence weather conditions like temperature and humidity. Here we show their joint variation over a growing season, and water use efficiency indicates how much water a plant needs to release in order to obtain a certain amount of carbon. Water use efficiency varies from subdaily to seasonal timescales. In this intensively managed critical zone, large-scale impacts result from small-scale processes. For example, photosynthesis and transpiration at a leaf level scale up to regional and watershed budgets of carbon, water, and nutrients. Critical zone observations at large and small scales help us understand complex feedbacks and processes that can help us improve our predictive models, our agricultural management, and environmental sustainability.